Imagine a man falling from the roof of a 42 stories building. How will it be felt? Dangerous. Hi, this is Saheli Chatterjee and I am going to discuss with you regarding such dangerous experiences when someone gets infected with viral hepatitis, as it is regarded as the 9th largest cause of morbidity rate worldwide. Liver is the largest gland in our body, and being so, it is obvious that it would account for an enormous number of chemical reactions and a horde of major functions. Some of the well-known among the lot are bile production, production of certain blood proteins, cholesterol, managing blood glucose levels, processing of hemoglobin, regulation of blood clotting, and a lot more. I would probably take a week discussing all its functions because the number sums up to more than 500. That's huge, isn't it? Therefore, it is worth discussing the major problem associated with this vital organ of our body. Hepatitis. Hepatitis can be defined as a state or collection of various abnormal physiological functioning of the liver that gives rise to inflammation in the gland. The World Health Organization has positioned chronic hepatitis as the 9th leading cause of death, affecting more than 3,000 million people worldwide. Although viral hepatitis is the prevalent cause of this disease, several other points leads to the occurrence of the inflammation, such as alcoholic hepatitis occurring in men and women consuming more than 80 grams a day and 40 grams a day alcohol respectively, consumption of various chemical agents like medications, supplements, abnormal immune response against liver cells, reduced blood flow to liver are some of the other reasons. The breakthrough in understanding the cause of this disease came in 1963 when Dr. Baruch Blumberg discovered an antigen that detected the presence of hepatitis B virus or HBV in the blood samples. Now let's discuss about the main causative agent, the hepatitis B virus. The hepatitis B virus, HBV, is a virus of the hepatinovirus family. HBV is generally transmitted between people through bodily fluids, such as blood, semen, and vaginal fluids. Dirty needles from drug use can also easily spread the disease. Unprotected sex is another prime way the virus can spread, again, through bodily fluids. While it might seem like a scary virus, HBV cannot spread through holding or shaking hands, sneezing, coughing, and so forth. Even sharing cups and utensils won't spread the virus. As long as someone steers clear of dirty needles and unprotected sex, and otherwise avoiding contact with bodily fluids, the risk of HBV transmission and infection is low. The hepatitis B virus consists of an outer lipid envelope and an icosahedral nucleocapsid core, composed of proteins having a virion diameter of 42 nanometers, making it the smallest animal viruses known. The nucleocapsid encloses the genome. This core contains a double-stranded circular DNA with 3.2 kilo base pairs and a polymerase which has reverse transcriptase activity. The viral genome encodes 4 overlapping open reading frames SCPNX. ORFS encodes the viral surface envelope proteins HBS antigen. ORFC encodes either the viral nucleocapsid HBC antigen or hepatitis B antigen. Hepatitis B surface antigen detects the actual presence of the hepatitis B virus. If a person test result is positive, then it is conferred that the person is infected with hepatitis B virus and is capable of spreading the infection through blood. The antigens are detected 4 to 10 weeks after subjection to HBV. If, after 6 months, the person still tests positive, his or her HBV infection is considered chronic. Hepatitis B A antigen is a viral protein made by the hepatitis B virus and is released from the infected liver cells into the blood. This test detects how much virus is in the blood as a result of very active viral replication. A negative test result indicates the virus may not be actively reproducing in the liver. In general, a person is considered very infectious when the test is positive, and less infectious when the test is negative. Liver cirrhosis. The infection with this virus can lead to infection ranging from acute to chronic. While both these states of the infection are associated with liver cirrhosis, well, in the acute stage cirrhosis is very minor or negligible as compared to the chronic stage. The liver may be injured by a single event as in acute hepatitis, or by regular injury over days as in chronic hepatitis, or by continuous injury as in daily alcohol abuse. Liver cirrhosis is a perplexed situation of the liver where hepatocytes are lost and irreversible scarring occurs which causes fatigue, itching, loss of appetite and yellowing of skin. In the normal liver tissue, the hepatocytes are arranged well with the intersecting triads that are associated with biliary ducts and blood veins. But with the progression of disease, the scar tissue increases quantitatively, causing the liver to shrink. The flow of blood to the hepatocytes decreases significantly, as the scar tissue presses against the blood vessels, and eventually, the cells die. Alcoholic and viral hepatitis B and C are common causes of cirrhosis which can be well detected by blood tests and can be further confirmed by liver biopsy using ultrasound and radio waves. Now let's discuss the replication cycle of hepatitis B virus. At the very first step, reversible and non-cell type specific attachment to cell-associated heparin sulfate proteoglycans. At this step, specific and probably irreversible binding to an unknown hepatocyte-specific pre-S1 receptor. This step presumably requires activation of the virus resulting in exposure of the myristoylated N-terminus of the L protein. Two different types of entry pathways have been proposed. 1. Endocytosis followed by release of nucleocapsids from endocytic vesicles. 2. Fusion of the viral envelope at the plasma membrane. Step 4 is followed by fusion cytoplasmic release of the viral nucleocapsid containing the relaxed circular partially double-stranded DNA with its covalently linked polymerase. At this step, transport of the nucleocapsid along microtubules occurs followed by accumulation of the capsids at the nuclear envelope which facilitates interactions with adapter proteins of the nuclear pore complex. Step 6 involves release of rcDNA into the nucleoplasm, but the mechanisms determining the breakdown of the capsid and the release of the viral DNA genome is not known yet. Step 7 involves repair of the incoming rcDNA by completion of the plus strand of the rcDNA by the viral polymerase followed by removal of the polymerase from the 5' end of the minus strand DNA. Then the short RNA primer used for the DNA plus strand synthesis is removed. Please note that these steps are mediated by cellular enzymes. Here in step 8, cccDNA formation occurs by covalent ligation of both DNA strands. This cccDNA molecule is organized as a chromatin-like structure displaying typically like beads on a string arrangement consisting of both histone and non-histone proteins called minichromosome. In step 9, cccDNA utilizes the cellular transcriptional machinery to produce all viral RNAs which are necessary for protein production and viral replication. The host transcription factors generally gets involved and regulate this process and may modulate viral gene expression by interacting with the viral promoters of the 4 major overlapping open reading frames that codes for the formation of various parts. In step 10, all 4 major mRNAs utilize a single common polyadenylation signal. Then processing of viral ribonucleotides, nuclear export as well as stabilization of the viral RNAs occurs, which appears to be exclusively mediated by host factors. Step 11 is associated with translation of the pre-genomic ribonucleotide to the core protein and the viral polymerase. Then regulatory X protein and the 3 envelope proteins gets translated from the subgenomic ribonucleotides. Now at step 12, complex formation of the pre-genomic ribonucleotide with the core protein and polymerase and self-assembly of an RNA-containing nucleocapsid occurs. At step 13, reverse transcription of the pre-genomic ribonucleic acid followed by plus-strand DNA synthesis occurs within the nucleocapsid. Then maturation of the RNA-containing nucleocapsids to DNA-containing nucleocapsids within the cytoplasm takes place. Step 14 is having two consequences. Firstly, the DNA-containing nucleocapsids formed can be either re-imported into the nucleus to form additional cccDNA molecules. Or secondly, the nucleocapsids can be enveloped for secretion. Then the envelope proteins are co-translationally inserted into the ER membrane, where they bud into the ER lumen. Then these proteins are secreted by the cell as 42 nanometers infectious virions called Dane particles if they have enveloped the DNA-containing nucleocapsids before budding, and are released. This is the final step. In between, multivesicular bodies are formed. These multivesicular bodies are a type of late endosome containing internal vesicles, and are formed because of inward budding of the outer endosomal membrane. The contents of this multivesicular bodies gets degraded, via fusion with lysosomes, or released into the extracellular space, via fusion with the plasma membrane. Types. Hepatitis B comes with two variants, acute and chronic. Acute hepatitis B. Acute hepatitis B infection lasts less than 6 months. Your immune system likely can clear acute hepatitis B from your body, and you should recover completely within a few months. Most people who get hepatitis B as adults have an acute infection, but it can lead to chronic infection. Chronic hepatitis B. Chronic hepatitis B infection lasts 6 months or longer. It lingers because your immune system cannot fight off the infection. Chronic hepatitis B infection may last a lifetime, possibly leading to serious illnesses such as cirrhosis and liver cancer. Serological markers. Serological markers are present on different scores of progression of the disease state, hence they are identified for diagnosis purpose. Please look into the table. HBs antigen. HBs antigen are first detected during the incubation period of 6 to 8 weeks before the appearance of symptoms. It can appear as early as 2 weeks and generally disappears within 3 to 4 months after exposure. It is the most reliable and universal marker of HBV infection. In the carrier and chronic state it persists more than 6 months. Anti-HBs. The appearance of anti-HBs may take several weeks or months after HBs antigen clearance, causing a window period. It is regarded as best indicator of recovery and immunity to HBV. HBa antigen. HBa antigen appears within 1 week after HBs antigen, it lasts for 3 to 6 weeks and disappears before HBs antigen clearance. Its presence indicates a highly infectious state. Anti-HBa. It appears before clearance of HBs antigen. It indicates decreasing infectivity and is a good prognosis for the resolution of infection. IgM anti-HBc appears prior to symptoms, and are found in high titer for a short time during acute stage. It differentiates between acute and chronic hepatitis. Anti-HBc total appears 4 to 10 weeks after HBs antigen appearance, and can persist for years or for life, therefore it is a prominent marker of HBV exposure. HBV DNA detection of low levels by PCR allows diagnosis of acute, chronic infection, carrier state, and also monitors the response to interferon treatment. On suspecting a probable disturbance in the physiology of the liver, the first and foremost test prescribed by the physicians are liver function tests. Liver function tests, also known as liver chemistries, help determine the health of your liver by measuring the levels of proteins, liver enzymes, and bilirubin in your blood. Commonly used tests to check liver abnormalities are tests checking alanine transaminase, aspartate aminotransferase, alkaline phosphatase, total protein and bilirubin. ALT. ALT is used by your body to metabolize protein. If the liver is damaged or not functioning properly, ALT can be released into the blood. This causes ALT levels to increase. A higher than normal result on this test can be a sign of liver damage. AST. AST is an enzyme found in several parts of your body, including the heart, liver, and muscles. Since AST levels are not as specific for liver damage as ALT, it is usually measured together with ALT to check for liver problems. When the liver is damaged, AST can be released into the bloodstream. A high result on an AST test might indicate a problem with the liver or muscles. ALP. ALP is an enzyme found in your bones, bile ducts, and liver. High levels of ALP may indicate liver inflammation, blockage of the bile ducts, or a bone disease. Total protein. Total protein is a measure of ratio of two types, of protein, albumin and globulin. The fall in this ratio indicates disease condition. Bilirubin. Bilirubin is a waste product and is ordinarily processed by the liver. A damaged liver can't properly process bilirubin. This leads to an abnormally high level of bilirubin. A high result on the bilirubin test may indicate that the liver isn't functioning properly. Looking into bilirubin, two questions comes to our mind. How and what? Let's discuss the first question. How are they produced? Please look into the pathway and follow the steps indicated with numbers from 1 to 5. Please look into step 1. Production. About 75% of the daily production of bilirubin is done by normal destruction of circulating red blood cells in the reticuloendothelial system, and approximately 25% of the daily production of bilirubin is derived from sources other than senescent red blood cells, like non-erythropoietic component and erythropoietic component. Now the conversion of the heme moiety to bilirubin is done by sequential action of two enzymes, namely heme oxygenase and biliverdin reductase. Step 2. Transport. Bilirubin leaves the site of production in the reticuloendothelial system and is transported in plasma bound to albumin. Step 3. Hepatic uptake. Hepatocytes have a selective and highly efficient system for removing unconjugated bilirubin from plasma. The transported bilirubin bound to ligandine, a cytosolic protein, that facilitates transfer to the endoplasmic reticulum, the site of bilirubin conjugation. Step 4. Conjugation. Conversion of bilirubin to excretable monoconjugates and diconjugates is carried out primarily by the microsomal enzyme uridine diphosphate glucuronal transferase. Step 5. Excretion. The conjugated bilirubin is then excreted into the bile, and is then transported to the small intestine, where it is primarily further degraded by intestinal bacteria and excreted in the stool. Step 6. Enterohepatic circulation. Intestinal enzyme hydrolyzes the conjugated bilirubin and release free bilirubin, which is then reabsorbed and transported by the portal circulation to the liver. Now the second question: what are its consequences? Alterations in any step of bilirubin metabolism results in increase in bilirubin level in the body. This results in several disease conditions. Some of them are jaundice, alcoholic hepatitis, Gilbert's syndrome, etc. Imagine how someone will feel if found positive. Let's consider this example to interpret liver test results. In this table it is found that AST and ALT levels are low, indicating normal, but this is not the case. This indicates liver disease. The AST and ALT level is low because probably all the liver cells are damaged and there is no further intercellular materials left for release. Even the ALP level is high, which further indicates liver problem. The total protein level is also low. Disease category. A liver injury is considered hepatocellular if ALT and AST levels are higher than ALP level. It can be cholestatic, if ALP level is higher than AST and ALT levels. It can be infiltrative, if ALP level is elevated and AST and ALT levels are near normal. Does hepatitis B affect human-gender ratios? Yes, it does, as it is found that infection is much more prevalent among the males as compared to that of females. This could be because of alteration in protein like apolipoprotein A1 or A2, in the livers of infected male but not in infected female. Diagnosis is confirmed by means of confirmatory test. ELISA. In 1971, two Swedish scientists, Eva Engel and Peter Perlman, discovered ELISA. The full form of ELISA is enzyme-linked immunosorbent assay. ELISA is a plate-based assay technique designed for detecting and quantifying substances such as peptides, proteins, antibodies, antigens and hormones. It comes with several variants. In this current study, Hep-ELISA kit is used. Hep-ELISA is a solid-phase enzyme-linked immunosorbent assay based on the direct sandwich principle. To perform this, at first, microwells are coated with monoclonal antibodies having high reactivity for HBs antigen. Then samples are added in the wells followed by addition of enzyme conjugate linked to horseradish peroxidase. A sandwich complex is formed in the well wherein HBs antigen is trapped, or sandwiched, between the antibody and antibody HRPO conjugate. Unbound conjugate is then washed off with wash buffer. The amount of bound peroxidase is proportional to the concentration of HBs antigen present in the sample. Upon addition of the substrate buffer and chromogen, a blue color develops. The intensity of developed blue color is proportional to the concentration of HBs antigen in sample. To limit the enzyme-substrate reaction, stop solution is added, and a yellow color develops, which is finally read at 450 nanometers spectrophotometrically. Let's look into the animated view of the overall process. Let's discuss another confirmatory test, PCR, which is not generally being performed due to its high cost. Prior to PCR, DNA needs to be isolated. For that, at first plasma is isolated by collecting blood from patient's serum. Then DNA is isolated from plasma by using series of steps. Then the purity of this isolated DNA is checked using a spectrophotometer, as it can able to determine the average concentrations of the nucleic acids DNA or RNA, present in a mixture, as well as their purity. For this, distilled water is taken as blank, and mixture of distilled water and DNA is taken as test. Note the volumes for both blank and test should be kept same, then the absorbance is recorded at 260 and 280 nanometers. PCR. The polymerase chain reaction was originally developed in 1983 by the American biochemist Karya Mullis. Using PCR, it is possible to generate thousands to millions of copies of a particular section of DNA from a very small amount of DNA. There are three main stages. Denaturing stage. During this stage, the cocktail containing the template DNA and all the other core ingredients is heated to 94 to 95 degrees centigrade. The high temperature causes the hydrogen bonds of template DNA to break and the two strands get separated. This results in two single strands of DNA, which will act as templates for the production of the new strands of DNA. Annealing stage. During this stage, the reaction is cooled to 50 to 65 degrees centigrade. This enables the primers to attach to a specific location on the single-stranded template DNA by way of hydrogen bonding. Only once the primer has bound can the polymerase enzyme attach and start making the new complementary strand of DNA. The strand's synthesis occurs in opposite directions. As a result, two primers, forward primer and a reverse primer, are used. Extending stage. During this final step, the heat is increased to 72 degrees centigrade to enable the new DNA to be made by a special tag DNA polymerase enzyme which adds DNA bases. It gets attached to the primer and then adds DNA bases to the single strand one by one in the 5' to 3' direction. These three processes of thermal cycling are repeated 20 to 40 times to produce lots of copies of the DNA sequence of interest. Let's have a look in this animation. Agarose gel electrophoresis. Arne Tiselius in 1931 discovered a method known as agarose gel electrophoresis. This method is effectively used for separation of DNA fragments of varying sizes. The DNA sample isolated previously is amplified by means of PCR and are subjected for separation to analyze the result. After electrophoresis, the gel is transferred to a gel dock for documentation. The gel dock is equipped with an ultraviolet light transilluminator, a hood to shield external light sources and protect the user from UV exposure, and a CMOS camera for image capturing. Now let's discuss the result. At first, a ladder is loaded into the first well to measure the position of amplification. Second, a known hepatitis positive sample is loaded which is showing amplification as it is positive. Third, test DNA collected from suspected patient is loaded and this is showing amplification, indicating the patient is positive for the test. Again, another test DNA collected from another suspected patient is loaded and this is showing no amplification, indicating the patient is negative for the test and have no presence of viral load. Thanks for watching. Kindly subscribe and share.
This video is brought to you by Science and Technology in association with Banerjee Infotech Solutions. For more assistance and help, kindly contact on the mobile number mentioned.